Well, thank you for this introduction and for inviting me here, and thank you for everybody joining. Kind of the talk is sort of designed as more of a high-level introduction to what we use artificial intelligence for in the context of de novo design at AstraZeneca, and kind of as illustration for this, I would use it in the context of REINVENT, so that would be the focus of my talk. So yeah, I'm aware it's probably a bit of a interdisciplinary audience, hopefully mostly around chemistry. But yeah, if you have more specific questions, I would be happy to answer them afterwards if you feel I haven't gone to the sufficient depth. And I do apologize, I don't have my camera on, but it seems that there is a bandwidth issue, so it's better this than risking to have a laggy connection. So I would actually, here give you a bit of an intro also to the department that I work for. Let me just get my pointer on. And here is me. And I'm not going to go through everybody here, just want to illustrate that there is a lot of young faces to our department. This is a lot of master students and PhD students and postdocs working with us and helping us to drive the science forward. And the head of the department is Ulla Enqvist. And we also have a GitHub page where we sort of expose our latest research and everything that is being approved and published as also a scientific paper. And I kind of suspect that if you've been attending previously other drug design related talks, you've seen that DMTA cycle, and DMTA stands for Design, Make, Test, and Analyze. And it seems like a very, very simple representation, but there is a lot of complexity behind it. There is a lot of expertise from different fields. There is, data scientists, machine learners. There is computational chemists and modelers, medicinal chemists, and all sorts of biologists from I don't know, structural biologists to assay biologists, and all these, they work together. They don't even sometimes know about each other, but it's like tens of people working this cycle, and that involves a lot of expertise, and it's kind of very expensive to do even single iteration of this cycle, and let alone doing that recurrently. And all this complexity of expertise and knowledge there kind of also offers a lot of possibilities for optimization, for making things better, for, well, in terms of optimization, maybe to speed up the way the cycle revolves, or synthesized, kind of improving the efficiency of iterating over the cycle. So all of this would be considered as some sort of a improvement of the workflow of designing novel drugs. And where we are focused, obviously, there is various areas. It's not just the designing part that can be optimized, but where we are focused is exactly on the design part. And our molecular AI department is working actually not exactly on supporting the projects specifically with design. We do that as well, but that's not our main focus. We mostly are into developing tools that are taking advantage of the advancements in the AI, and we use this to kind of help answer some major problems that are kind of related to the drug design. And I think the two main questions that are kind of popping out when we speak about drug design is like how to make, so what to make next as a compound, and how to make it. And these are two different branches that we're trying to address at Molecular AI. So reaction planning is kind of a topic that my colleagues are a lot more competent. So you may pick some of the faces from the previous slide to give you a much better presentation on that topic. So I'm going to leave that out, but it's roughly about retrosynthetic planning or condition prediction, or helping in various ways to the medicinal chemists to plan how to make the compounds that are proposed from the de novo design tools. However, yeah, my focus is mostly on de novo design tools, and kind of before diving into the specifics of the application that we are developing and maintaining at Molecular AI, I would probably give you first some kind of abstract generalization of how I see all the de novo design tools from the beginning, I don't know, from 30 years before till now. I would actually generalize them as 3 major components. So in each de novo design tool, we have a search space, and we have a search algorithm and a scoring function. And the search space can be just as simple as a static database, right, with compounds. There's not much novelty there. It's constant, it's there. Search algorithm could be just a brute force enumeration of all the compounds that are in the database, and the scoring function is something that sort of reflects your project objectives, whether it's going to be activity, selectivity, permeability, or I don't know, whatever other properties you want to put in there. They are objectives that your compound, your potential drug candidate should be able to meet. And until recently, at least that's my my viewpoint, the state of the art was to have, for instance, static database that is maybe made out of fragments, and you can have a genetic algorithm that is making, helping to make some clever choices and combine these fragments into molecules. And you have also the scoring function that is evaluating the resulting compounds. And what we have sort of adopted as a new approach, and what also the tool that I'm kind of presenting today, REINVENT, is using, is sort of replacing the search space, the chemical space, with a generative model. And just to be clear, there is no fragments there or anything like that. There is just tables with numbers, and it's just probabilities of certain tokens to be generated, but I'll get to that. And we also replaced the search algorithm with a reinforcement learning algorithm, and the scoring function just remains the same. So the objectives don't really change there. And also want to give you a bit of a feeling of, you know, when I speak of a chemical space, what is kind of commonly understood. So we have this kind of a, maybe it is a speculative number, but it's a really large number of what the potential chemical space is. And maybe some of these compounds from this chemical space are never realistic to be synthesized. Maybe some of them are synthesizable, but we just don't know how to get there. And what we kind of currently estimate that we have as a chemical space coverage is probably 1 tiny pixel, actually, if we are to talk about up to scale about what is currently available. So we really have access to a negligible fraction of the chemical space, and relying just on databases to explore that chemical space seems a little bit limiting, and that's why I kind of speculate that generative models tend to hold more potential there. Anyway, so in terms of, you know, all the entities in the chemical space, we all know, you know, the good old chemistry classes and how we've studied chemistry. We always represent the compounds in this kind of a 2-dimensional graph representations. And that's how we are used to think of chemistry, which, you know, it's not necessarily the most accurate representation of a molecule, nor the most exhaustive, but it's something that we are used to, and that's how we mostly operate, and that's how we mostly work with. And you know, to the ones that are computational chemists, I guess they're very well familiar with the SMILES format, but just to explain it here, you can see the color mapping between the different nodes of the graph here, how it maps to the SMILES string below. It's one of the many formats under which we can also represent molecules. And SMILES, in a sense, it's actually an implicit graph if you think about it. So it's a graph mapping, but it's a string representation. And one of the few kind of specific properties for the SMILES representation is that if we, for instance, start the count of this graph from different point, let's say from here, will result in an entirely different SMILES string, which is again representing the same molecule. So if we think about it, we actually have numerous SMILES representations for this molecule, and we can say that it's actually a very, very convenient feature because it can serve also for data augmentation. We can represent one molecule with multiple SMILES strings. And well, why using SMILES format? So I should clarify, there is also generative models and deep learning techniques that rely on graphs, but we are kind of focused on the SMILES that has worked for us the best. And for the purpose of this talk, I would only focus on the smiles representation, and that's what reInvent uses. So why we use smiles is because we've borrowed architectures from other fields, from natural language processing, and they work very well with text representations. So what we've done here is to actually train the model to understand the smiles syntax. Actually,
Atanas, we have a small connection problem. We have a small lag here. Yeah, there's a small connection problem. Atanas will try to reconnect.
give you some intuition about how it works. So
Atanas,
here
sorry,
you have a little bit. Yeah.
you broke up. So could you please
Oh,
maybe
sorry. Where did you lose me?
like 1.5 minutes ago?
Oh my God. Oh, all right.
I'm
That's very
sorry.
bad. Oh dear. All right. Sorry, guys. So did you get this slide at least?
No.
Oh right. Oh my God. This slide?
Yes, this was completely fine. I think this was finished already, more or less.
Okay. All right. So I was just explaining how the tokenization of the smile strings is done. So this is an example where we have some molecule, and this is its corresponding smile string, and this is the one hot encoding. And essentially the message here is that the generative models don't really see the smile string, but they see its one hot encoded version of the smile string. And we have here vocabulary, and that smile string is mapped by the vocabulary into this two-dimensional matrix. All right. Maybe check once in a while with me if I'm still online. All right. So here, this slide illustrates how the generative model produces the smile string. And you can see here, this is another vocabulary, and you can see the different probabilities for each of the tokens to have been to be generated by the generative model. And the darker the color, the higher the probability. And here the structure will appear, and underneath the smiles will pop. So for the first step, the carbon has the highest probability, but it actually picks an oxygen. So it's a stochastic process. It can also generate the end token, and then it will just end very quickly. So you can see here the oxygen being generated. And as the next step, the probability is also for the double bond to be generated, and it picks that one, and so on and so forth. And it generates, here you can see the structure emerging, and here you can see the smile string being generated. So hopefully this gives you some kind of an intuition how, based on the probability, different tokens are generated by the generative model, and that results into some kind of a molecule from the deep learning model. Right. So going now to reInvent, as I mentioned before, reInvent is a deep neural net that has been shown the chemical data, a lot of smiles representations, many times, many epochs, and it has learned the smile syntax, and it has learned it to generate smiles with very high validity. And that's all it knows, right? This is the concern of the model, just to generate valid smiles. It's like 99 point something % validity. And the next thing we do usually with this generative model is we put it in some kind of a learning scenario, right? So we give it some objective, could be a combination of various predictive properties or something else. We just refer to it as a multi-parameter optimization score. And what the generative model does, it generates a batch of molecules, then they're scored, then that comes back as a reward, and it learns from what's been propagate it back, and it generates a new, better batch of molecules. This improving gradually the ideas that it generates until it starts producing something that is of value and that we would actually collect. To give a further intuition here, I illustrate with just a really toy example where REINVENT generates structures that are similar to selectivity. And let's see if this animation will play. Or maybe I just paused it. Bear with me. Okay, right, started playing. So yeah, the objective is selectivity, and at the beginning you can probably see it just starts from random structures, and at some point it will start picking patterns here, and it will start generating molecules. You know, it will retain the pattern, and it will start generating molecules around this pattern. You'll start exploiting it, and until ultimately it generates only selectivity or very, very close analogs. So this is obviously just a toy example, but it's intended to give kind of an idea to the people have not encountered, you know, haven't dealt with generative models before, so you can have an understanding. All right, so we usually utilize much more complex scoring functions. So this is just to remind you about the three components: the generative model, reinforcement learning, and the scoring function. And the scoring function is actually what I'm going to be talking about next. So like I said, you know, this was just a very simple example where you have similarity criteria to some concrete molecule, but you can have a list of molecules, or you can have scaffolds, or you can have also dissimilarity criteria. So you can have criteria to generate compounds that are as far as possible from certain list of compounds, or you can penalize for certain type of undesirable groups in your molecules. So you can, you know, penalize for certain moieties. This is completely valid. You can also use predictive models, regression and classification. You can use various calculated properties, whatever you can come up with, molecular weights and TPSA, etc. You can use also shape similarity. So this is a bit of a 3D information, pharmacophore shape similarity. You can use it the same way you use as you know, the 2D similarity criteria, and obviously you can use docking. So anything that provides a score can be actually propagated down to a scoring function and then to the generative model. And here, this is just a schematic representation. So agent stands for the generative model that I'm talking about. It's in the reinforcement learning scenario is referred to as an agent, and the agent generates smiles or takes an action. Here there is a score modulating block that results into some score, and it goes back to the agent. And I guess you can devise here the learning scenario to your liking, but what usually we have as a special component out of this score modulating block is this diversity filter, which allows us to balance between exploration or exploitation, depends on the case that you have. And you know, compounds that are generated during the reinforcement learning are actually collected in the diversity filters memory and then recorded as a output from the entire reinforcement learning. So this is how we collect the ideas. We don't do it after the reinforcement learning. After the reinforcement learning, usually the agent is discarded. So speaking of exploitation, exploration, just to explain in which context you might want to do that. So exploitation is when you're already like in a good place. You have your series of compounds and you just want to optimize certain properties, maybe reduce toxicity, or I don't know, maybe make it a little more selective. So whatever it is, it's aims at retaining the main scaffold, so kind of staying in the same place of the chemical space the way we perceive it. You either escape maybe a pattern space, or maybe you'll want to go to different type of scaffold. So essentially it is for scaffold hopping, where you want to go to different types of molecules for whatever reasons. And this is actually what impresses humans the most, because this is very difficult for a human to do that. And that's what is kind of they see as the highest value for using the generative models. There is also one extreme case which I don't recommend getting into. It's mode collapse, where when your kind of generative model starts producing the same or similar compounds all the time, it's just wasteful. You just need the compound to be generated only once. That's it. You don't want to generate it 1,000 times. Right. So I have a couple of examples that are revolving actually around scaffold hopping, because I said that this is, it's my impression, this is the most impressive part. And these are just examples, actually really fresh out of the oven. It's manuscript that hasn't been yet released, but it should be out sometime in July. So Christian, Margaret, and Jeff Gu shared those with me. And so here the target is kinase, and we have some kind of a reference structure, and we use docking as a scoring objective. But it's not only docking, there is also a bunch of other supporting components that we use in the scoring function. So we want, for instance, to have the molecular weight constraint, because docking has, well, all the docking scoring functions tend to have this tendency towards rewarding larger molecules. So for instance, that's one way to penalize it. We have also other components because they, for instance, prevent from exploiting other weaknesses of the scoring function of the docking software. So that's, there is a lot of interesting examples there that, for instance, it can start generating very high polar surface simply because the scoring function is rewarding this. So reinforcement learning will exploit these kind of weaknesses in your components of the scoring function, right? And here there is an example where it proposes a ligand that, retains the desired binding pattern, but it has an entirely different scaffold. So this is just a single molecule example. I have another example, for instance, where I can we're trying to illustrate how it generates very diverse data sets. So it's a different run, it's a different target as well, COX-2. And here we have known active compounds and we have known inactive compounds. And here from the docking run, the resulting data set has produced some compounds that are similar to the active compounds, right? Over here, this is a Tanimoto similarity measurement. And we have some compounds that are similar, but the majority of the compounds are entirely different scaffolds. So here you can see something that is very similar to the non-active. And at the same time, it has realized what is not active. So it hasn't generated anything that is similar to the decoys, to the inactive compounds. So you can see that from this bar plots. And here there is another example of reinforcement learning that can. So the message here that I'm trying to convey is that actually reinforcement learning can take you in any possible direction, maybe even regardless of the state of your agent. Well, given that it's sufficiently healthy, it's not producing too many invalids, or it has other issues. But if you look at this, these black dots here, so this is a UMA plot. Here is the black dots are representing compounds that are generated by untrained prior state of the generative model. And it's just random, but it's kind of clustered still in some way. And then after we subject this generative model to reinforcement learning, you can see it's 3 different runs, but it's with the same scoring function. And from 3 runs with the same scoring function, we can see that we generate quite different populations of compounds. So even one run with another can lead in a different direction. It really depends on how you set up your scoring function objectives. I don't know if I have the time, but I would like to mention a few words about another kind of component to REINVENT. So this was a separate publication. It was it's currently under review and the preprint is out. And the point, the purpose of this libinvent tool is a little bit more specific rather than the general purpose of REINVENT. So libinvent is focused on creating molecular libraries, so focused molecular libraries. And it has the same setup, like generative model, reinforcement learning, some objectives generates the library. And what I mean by a library, so try to give you my definition. So this is a set of compounds that share the same scaffold, and they can be generated under the same conditions with the same or very similar types of reactions. And the point is for batch synthesis. So you can use these molecular libraries for batch synthesis, maybe 96 well plates. And you can, in this case, you need to have the same conditions to carry on the reactions. And here is just an example of a scaffold and some decorations or moieties that are specific for the individual attachment points, and then that will result into a new compound. So this is actually a continuation of a previous work from Joseph Arispus, where kind of the model is borrowed from there, where one could actually provide a smile string with attachment points, and then the generative model would generate the moieties that are attached to these attachment points. And the learning scenario, probably by now, it's painfully familiar. It's again reinforcement learning. We have the input to the agent as a scaffold. There is a, the generative model. The agent is generating the smiles, then they are scored, and then it learns from that. The difference here is, oh, I have an animation. So the difference here is that we also have something that is called reaction filters, and we add this to the scoring function. So we sort of can enforce a specific type of reaction to be applied to the specific attachment point. And I think here you can see that better from this side. So I even have a few examples. So what are the goals is to have, on top of your learning objectives, the reaction preferences, and you can generate your data set. And what we also realized in the course of work is that we also need to maybe hook it to one of the reaction prediction tools, so that it also helps us to identify easier the building blocks that come out of REINVENT that are suggested there, so we can make the life easier for the synthetic chemists and to help them kind of compile their library and go for the batch synthesis. All right, so I have a couple of slides for toy examples. And this is the scaffold that I will be kind of illustrating this. It's borrowed from a DRD2 data set. And the objective here is to use different reactions for the 2 attachment points that are illustrated here. And the concrete example is to use kind of as an objective some pharmacophore similarity. So we can have this as a scoring function component, and we can have this, for instance, reaction filters there, like amide coupling and Buchwald-Hartwig for the 2 endpoints. And here you can see what is the outcome. So the amides are generated on the corresponding end, and the Buchwald-Hartwig reaction is kind of, these moieties are compliant with the Buchwald-Hartwig reaction. And I don't know, if you're skeptical, maybe we can also show you some other example where instead of amide coupling, we use sulfonamide coupling, and it generates another batch of results where sulfonamides are attached to the corresponding end. And these are like the high scoring compounds, so maybe there is certain similarity also due to the a little bit more narrow space from the pharmacophore objective. All right, so I'm kind of near to conclusion here, and here is the GitHub repositories. These are freely available. You can play around with it. They're mostly computational chemist friendly. They're not very medicinal chemist friendly at this stage, but I'm aware of people building interfaces. We also do that. Even as computational chemists, we want to use it behind an interface rather than to deal with the complexity of artificial intelligence. And maybe just as one final kind of a message from me, it's kind of not always sufficient in order to change the people's mindset and to kind of break their skepticism to only do the impactful scientific advancements. It does require a little bit of GUI development and having all that complexity wrapped behind an interface. For instance, Solve IT are pretty good at this. They have amazing GUIs. And there is also the necessity really to sort of change the mindset of how to work with these tools, because until recently, we were still kind of used to have the responsibility for proposing the compounds ourselves. But here with these tools, it actually changes the paradigm towards having to ask the right question, really. You just have to ask the right question from the artificial intelligence, and it will give you all the answers. So that's from me, really. Thank you.